So hello everyone, and welcome to the second in this series of seminars co-hosted by the BSA Science and Technology Studies Group and the New Materialisms Study Group. These seminars explore perspectives from scholars working across and between these schools of thought to ask how conversations across STS and the New Materialisms might enrich, but also interrogate the others' approaches, methods, and practice. So the seminars are hosted by the BSA New Materialisms Study Group conveners, Pam Aldred, Faye Dennis and Nick Fox, and the STS group conveners, myself, Emily Ross, and Julia Swallow. Eva Giraud gave a really brilliant seminar last month, and that's now available to view on our study group websites. And today we're absolutely delighted to be joined by Professor Deboleena Roy. Professor Roy is Professor of Neuroscience and Behavioural Biology and Women's Gender and Sexuality Studies at Emory University, Atlanta. She holds a PhD in reproductive neuroendocrinology and molecular biology and uses this experience to create what she calls new zones of proximity between laboratory science, molecules, and feminist political landscapes. Her beautifully written monograph, Molecular Feminisms, Biology, Becomings, and Life in the Lab, was published by the University of Washington Press in 2018. And in this book, she uses her interdisciplinary approach to develop new perspectives, vocabularies, and tools to approach biology from outside the lab. Her work has also featured in publications including Signs and Catalyst, and in many scientific journals too. So in this session today, Professor Roy will discuss J.C. Bose's notion of a physiology of response and how this might help us to reimagine feminist encounters with biology. She'll talk for around half an hour, and then we'll open up to questions. And this part will be led by my co-chair, Nick Fox. Do you want to say hello, Nick?
Hi.
Hi. And in terms of the Q&A, if you would like to pose a question to Professor Roy, please use the chat facility to write question, and Nick will invite you to speak. And we'll do this once Professor Roy's talk is over. So I'll now hand over to Professor Roy to present her exciting talk, Microphysiologies of Desire and an Ethics of Matter. Thank you.
Thank you so much for the introduction, Emily. And I also want to thank Faye Dennis for inviting me to speak. It was a lovely introduction. And, you know, I'm always so grateful to hear that people have found the book and that someone's read the book and then, you know, can even quote a little part of the book. So I'm really honored to be here and to have this chance to speak with you all. I also want to thank Nick. Nick is going to be moderating the questions at the end. I do hope that we have a chance for some rich discussion. I'm going to present, Faye told me to present for about, 25-30 minutes. And I'm really looking forward to some rich discussion afterwards. So I know that the theme for your talk, for all of you, you've been thinking about new materialisms and the intersection with STS. Over the years, I've actually been contacted by different groups of thinkers and knowers to think about the intersections of new materialism and STS. And by this, I mean, there are students working in a lab who have never read anything about new materialisms, but they're asking questions that to me sound like they are, questions around new materialists and feminist theory. I get questions and chances for collaboration with artists who are thinking about these questions, philosophers and, sociologists. So, I really realized that I have created some kind of practice of making these connections. And I think the book really is about that, that practice. So the talk that I'm going to share with you today is really kind of an overview of a lot of the work that I've been doing, but the questions that I've been asking over and over again in multiple different ways. But they originate for me as a feminist scientist who worked at a lab bench. I've been away from the bench for some time now. After that PhD and a postdoc, some of those questions and kind of the pauses that I bring up in my book around working with cell lines, about working with molecules, made me go down this road of exploring feminist theory. And to do it seriously, I realized I actually needed to kind of do another PhD, which I didn't do a PhD again. What I ended up doing is becoming a tenure track faculty in a women's studies department and having to, learn the field as I got tenure. But those questions that originated in the lab have stuck with me. And so I'm going to be sharing most of those now. Now, I'm not sure about where people are coming from and, their kind of background and the terms and how I'm using these terms. I wanted to kind of share that with you all. So I use, obviously, the term feminism a great deal. The book is called Molecular Feminisms. For an American audience, often, I have to explain, well, what do I mean by feminism? And a lot of the feminism here is really around kind of liberal, humanist, equal rights kind of feminism. So I like to explain that the feminism I'm talking about, that I like to think about, actually is like a toolkit. And it's a toolkit that I use to think about questions around norms, what gets established as a norm, and therefore what gets marginalized when you're creating a norm. So, you'll see, for those of you who are familiar with queer theory, that there's an overlap here between the feminist theory and queer theory. But to me also, feminism was a way to think about difference, really, not just in terms of one's sex or one's gender or one's sexuality, but also in terms of one's race, class, ability. Feminism was that kind of toolkit, as I say, to think about these questions of difference in a more meaningful way. Molecular, the term molecular, I use that a lot also. And of course, I'm playing on the term molecules and the molecular level. But I'm also here referring to how a lot of feminist and STS scholars and feminist STS scholars have also used the term, drawing from Deleuze and Guattari's work, to describe minoritarian projects and a type of minoritarian politics. So the title of my talk today, The Microphysiologies of Desire, that's a direct kind of draw and homage to the micropolitics of desire that Rosie Bradotti's work looks at, that Elizabeth Gross's work has looked at, and the terms that they borrow from Deleuze and Guattari as well. Becoming. This maybe also needs no explanation to this crowd, but I think for a lot of, because my work is interdisciplinary, walking people to this idea of becoming, I do sometimes contrast it, sorry, with this idea of being. And the way that, a lot of my undergraduate students in the neurosciences or molecular biology, how I bring this to them is, I think, when you think of a molecule, is its identity fixed? Is that how we treat biological matter? Or are we interested in what that matter does? Kind of the signal transduction pathways of that molecule and that matter. And if you can think of movement, if you can think that, an identity or even like a material identity can change, that's the idea of becoming. So it's this concept that emphasizes the capacities of flux and change, where the properties and components of nature are understood in terms of processes and events instead of an identity. So those are some kind of just key terms that I want to share with you all as I launch into the rest of the talk. So maybe also, since this is the Sociological Association, you all will be aware of this. But, as a feminist neuroscientist and a joint faculty member in the Department of Women's Studies and Neuroscience and Behavioral Biology at Emory, interdisciplinary work has been my bread and butter. And after years of conducting research and teaching courses across the natural sciences and humanities, I do know that interdisciplinarity can be thought of in multiple ways. And so the primary focus of my interdisciplinary work has been motivated by a commitment to understanding and learning to appreciate the intricate practices of disparate disciplines. And this may be due to the fact that I began my training as a scientist and began training to become a feminist scientist during a time that, we now refer to as the science wars. And I think that this crowd must be familiar with that. But for those of you who might be unfamiliar with the science wars, this was a period of heated debate and somewhat hostile exchanges, I think we can say that, between scientific realists and post-structuralists that took place in the mid to late 90s. These debates involved discussions around scientific objectivity and the possibilities of societal influences on scientific inquiry, which we might today, and we hear this all the time, refer to as unconscious bias. So the publication of several books and journal articles during this period led to a series of high-profile disputes, and in some cases, a deep mistrust between academics who are in the sciences and those who are in the humanities. So as a graduate student in the neurosciences in Canada, I started my PhD in molecular biology and reproductive neuroendocrinology just as the science wars were in full swing. I was actually blissfully unaware of one of the most scandalous scholarly disputes that transpired during the science wars, and that's the so-called affair. But somehow I had already come to the understanding that science and society were co-constituted. I'd never taken an STS class. Actually, I'd never taken a women's studies class, but somehow that made a great deal of sense to me. And I already knew that I wanted to become a feminist scientist who could bring reproductive justice work into conversations with reproductive biology research. That just made sense to me. And it could be that I grew up in Toronto, and at this time in the early 90s, there was definitely a lot of activism happening in the streets of Toronto. The University of Toronto is located right in the heart of downtown Toronto. So it was hard for me to divide those worlds of activism and learning my academics. So becoming this feminist scientist who could bring reproductive justice work into reproductive biology research, I knew that it would involve learning the intricate practices of different disciplines and developing an appreciation for different ways of encountering the world. So, this happened. Imagine people in a crowd like this, but this was back in 2019, before the pandemic. And so this was after 2 decades after the science wars. I do think that we have found ourselves in this new era. We find ourselves in a political time and place where all forms of academic research, whether it's in the physical or natural sciences or the social sciences and humanities, are all potentially under some kind of threat. And who would have thought that back in the 90s, when the science wars were raging, that scientists would have to stand up for science itself as well? Who would have imagined that scientists would be marching for science? And it turns out that the skills that a lot of humanities scholars and social justice advocates have had to hone over the last 2 decades, including interdisciplinary institution building, community organization and outreach, and finding creative ways for obtaining funding research and support, they are now key skills that's required for everyone in academia, scientists included. So in a curious way, I do see this strange moment as providing us with opportunities of forming new interdisciplinary alliances, such as the one that you are trying to bridge between new materialisms and STS work as well. So my book, Molecular Feminisms, Biology, Becomings, and Life in the Lab, it's really a project about learning how to make interdisciplinary connections. And it's written not so much as a treatise between following the science wars, but rather as a guide for building coalitions and for creating not what I refer to, what others refer to as horizontal social movements from within the sciences and the humanities. The book aims to provide a reflective space for both feminist scientists who are in the natural sciences, who participate in bench research, as well as feminists who are in the humanities and social sciences, who are eager to use scientific research and data to inform their feminist analyses. It also invites scholars of feminist theory, post-colonial studies, philosophy of science, animal studies, post-humanist ethics, and the biological sciences to participate in joint conversations and create shared vocabularies for creating interdisciplinary work. And the project really began with the scientific research that I conducted on the molecular effects and signal transduction mechanisms of melatonin, estrogen, and androgen receptors in an in vitro model of hypothalamic gonadotropin releasing hormone neurons, or GnRH neurons. So I'm going to talk a little bit about that research and what kind of motivated me to start asking some questions that now, has brought me to these areas of new materialism and STS. So my work in reproductive neuroendocrinology and molecular biology revealed the presence and mechanisms of feedback regulation by the pineal and gonadal hormones at the level of the brain and the hypothalamic pituitary, or the HPG axis. These findings were significant for two reasons. They provided evidence against a top-down or hierarchical paradigm that was commonly used to study the HPG axis in mammals. So, the idea is that the brain gives all the signals and is the control center, and it sends a signal to the pituitary gland to release luteinizing hormone and follicle stimulating hormone, which in turn, travel to the ovaries and to the testes to turn on the synthesis of estrogen and testosterone. Well, the idea that this was kind of a hierarchical model was being questioned, but what was not there was proof that there was actual feedback happening at the level of these GnRH neurons in the hypothalamus. So my work was able to show that. It also, one of the, I think, significant findings from this research is that they showed that hormone-based contraceptives and hormone replacement therapies, or HRTs, may not only act at the level of the ovaries or the testes of the uterus and the breast tissue, for example, but they might actually have broader systemic effects, including neuroendocrine regulation. So I was able to show estrogen receptors in the brain, for example, and I'll talk about that experiment a little bit more. But the fact that there are receptors for estrogen in the brain means that any kind of HRT is going to have some effect at the level of the brain as well. So I'll go to the next slide here. So interestingly, my decision to pursue this scientific research in the first place was in fact driven by my involvement in reproductive justice movements, both at the local and national levels in Canada. The challenge I faced, though, once I got into the sciences, that was in order to get to the important findings that related, that was important to reproductive justice work, my research required using molecular biology techniques and practices that many feminists also harshly critiqued. And these practices in turn included the use of bacteria, transgenic animals, in vitro cell lines, recombinant DNA technologies, and so forth. So it became clear to me that for those of us who were out there who wanted to become feminists, but were also wanting to participate in the creation of new scientific knowledge on the body at the molecular and genetic levels, and work with the matter of biology, facing such dilemmas was going to be an important part of the project of diversifying science. And it was going to require a toolkit. And this is that feminist toolkit that I refer to. So since then, the dilemmas that I've encountered in trying to practice science as a feminist and as a feminist of color have been challenging, but also extremely productive. They've provided me with a skill set that has been born out of trying to create these interdisciplinary research discussions and collaborative methods. And as a result, my efforts and contributions have been focused on creating these kind of sustained dialogues around questions of matter, questions of ethics and difference at the intersections of molecular biology research, neuroscience research, and reproductive justice work. So I'm going to take you now to kind of one of the motivating factors of kind of what brought these questions together for me. My parents are from India, they're Bengali, and they migrated to Canada in the late 60s. And, growing up, I was born in Canada. Growing up there, they did make sure that I learned as much as I could about my Indian heritage. And one of the points of pride, since I did have an interest in the sciences and showed from very an early age on that I wanted to become a scientist, they would tell me stories about J.C. Bose, and maybe some of you know about his work already. But Jagadish Chandra Bose did some really amazing experimentation in the late 1800s and early 1900s. And here's a picture of him giving a talk at the Sorbonne in 1926. And the title of his talk was on the nervous system of plants. And we can get into this a little bit more in the Q&A if people are interested. But what J.C. Bose did, and I knew about this experimentation from a very young age, is that he was trying to look at whether plants have a type of response system that we as humans think we only possess. This idea that response, or wherever what comes from that, the idea that a non-human entity can also have a way of sensing, a way of interpreting, a way of reacting, and it's just not simply like a physical response, that there's something else beyond that. That really intrigued me as a kid. And in fact, there was a film put together. Stevie Wonder did the soundtrack for this film, or I think his album came first, called The Secret Life of Plants. And in that, they have a film around it. They talk about J.C. Bose's work, where he put two plants beside each other. And if any kind of damage or, was done to one plant, the other plant would actually respond. And this was an apparatus on the right here that Bose used to actually measure that response in both the living and non-living. So I was just fascinated by this as a child. And so I brought this with me, I think, when I started studying molecular biology. I've always been interested in, so I did an undergrad in microbiology. So I just think the world of going kind of smaller and smaller drew me into asking, like, how do we follow this idea that response can come from anywhere? Can we even see it in molecules? So some questions that, were posed as I tried to do this research, but also aware of the science wars and all the critiques of science, is how do we continue to do science after those critiques? How do we work toward a biology that we desire? And here, this question of that we desire, I talk about, and this came from my training as a faculty to teach women's studies classes. I had to learn the work of Freud. And that famous quote of his, "Anatomy is destiny." I've thought that we need to hold on to anatomy. We don't just let go of it, but is it our destiny? Can we make anatomy part of what we desire? And so another question was, how can we bring questions of context with us when we do encounter matter? So as we, as I walk into a lab and put on that white lab coat, but you can see in the back here, maybe I always wore a red lab coat. I didn't like the white ones, but how do we bring that context in with us into that lab space so that the questions we pose and the partnering that we do with the molecules and the animals and the cell lines, we can bring some of our hesitations with us in those questions. How are we to encounter matter? This is a question really about that relationship, and it goes along with that last question here. How do we reconfigure the relationship between the scientific knower and what is to become the known? Is how do we think about objectivity? What do we do when we think we can be purely objective? What kind of distance do we think we've created between who is the knower and what has become the known? These are questions that I think sat with me, but I didn't know that other people were thinking about this until I came across the work of Barbara McClintock and her, it's written by Evelyn Fox Keller, but that feeling for the organism, this idea that Barbara McClintock had because of the years of research that she did on the cytogenetics of corn. She worked at Cornell and she would go into the fields, she would bring the maize into her lab, she would crush it up and she would look under the microscope, and then for hours, spending time with the material, she would find herself, there's this beautiful line in the book that she would find herself right inside with the chromosomes. And that's how she discovered transpositions, the actual jumping of genes. So that kind of intimacy that you could actually develop that as a scientist and have that orientation when you do encounter matter was something that was very appealing to me. So this is not something that I'm going to talk too much about, but if you had a chance to look at the book Grass and How Grass Grows, has a kind of an important role in the book. And this idea of this kind of ontological univocity, these ideas of horizontal social movements, to me, actually watching grass grow, which I did for hours on end a few years ago, really kind of taught me lessons that grass has a lot to teach us. So just as feminism is a type of toolkit, I think grass has a strategy, and that we can all learn from what grass does. So I won't go into this in too much detail, but you can see here that, you know, there are stolonic runners that get thrown out, and you know, these different kind of clonal populations of grass learn to join and make connections. And I think that the project, I use this metaphor to think about how to make connections. So this last part of this talk, I'm going to talk about the actual experiment that got me to some of those questions, and hopefully we'll open up kind of the theoretical parts of the kind of the new materialist work that has influenced me. So at the beginning of my doctoral work, I had the opportunity to find new forms of communication between GnRH neurons, estrogen receptors, and estrogen molecules. The molecular biology techniques that I was required to learn in order to conduct my experiments taught me how to face the lab bench, how to form experimental togetherness around shared objects of perplexity, and how to break bread, as it were, with my scientific peers and colleagues in the lab. I long to create new zones of proximity between molecules and my own feminist political landscapes by examining a biological interaction and process that had been marginalized in the sciences, but was relevant to both molecular neuroendocrinology and women's reproductive health. So I like to say that I was after the molecular within molecular biology. Those are the untold stories, the narratives that had not yet been kind of explored enough. So that's my excitement when one of my first experimental research tasks involved searching for the expression of estrogen receptor genes and the synthesis of estrogen receptor alpha and estrogen receptor beta proteins in the in vitro cell line model of GnRH neurons known as GT17 cells. So this is just an image that I took years ago, immunocytochemistry image, using fluorescence techniques, and these are the actual GT17 neurons. And these neurons actually came from a mouse model that was produced so that it would have a tumor in its hypothalamus. And so the GnRH neurons incorporate the in vitro ones that I used, incorporate a gene that causes a tumorigenesis. And so this is why they could survive in vitro. So, and GnRH itself, that's produced by these neurons, it's known to be the central hormone, as I explained earlier, in the HBG axis. And it helps to regulate, you know, that secretion of LH and FSH, and ultimately estrogen and testosterone. But it was through this in vitro model of GT17 neurons that I was able to find that estrogens and androgens actually feed back at the level of this neuron itself, and that these neurons express those receptors. So in this article that was published a few years ago, that's when a long time ago, actually, no, 2 decades ago, that's where these findings were shown. And so my doctoral supervisor at the University of Toronto, her name is Denise Belsham. She was interested primarily in examining the role of estrogen in the brain in its reproductive capacity. And estrogen itself is a very interesting hormone. It's been shown to negatively regulate GnRH synthesis, but it's also necessary to introduce a pre-ovulatory surge of GnRH. So estrogen does 2 things. And just this here, that it's not a stable molecule, that it's doing multiple things. This was intriguing to me. So estrogen both suppresses GnRH secretion, but you get a spike in estrogen and it enhances actually GnRH secretion during the menstrual cycle. So one would imagine that estrogen would have something to do with the regulation of reproduction at the level of the brain. But the research that we conducted at this time, it was kind of thought of as being unnecessary because other scientists in the field had decided that, you know, GnRH neurons don't express estrogen receptors, but that there are intraneurons that are doing this work. And so that was what we were up against, that we had to show that these neurons themselves could be impacted directly by estrogen. So I was working kind of athwart to expert knowledge in reproductive neuroendocrinology that was unconvinced of a certain trait, and unwilling or uninterested in spending time designing experiments to recognize these new zones of indiscernibility. So scientists at the time had, dismissed this part because they thought that, other parts of the brain might be impacted by estrogen. And they didn't even think that estrogen maybe really was present, that it was kind of, what are the minoritarian kind of actions of estrogen. So at that time, estrogen receptor research was full of, it was really kind of interesting as I started my PhD, because they realized that it didn't just come in this one form. There was a chance that there was a second type of estrogen receptor. And so just before I started my research, this kind of stable unitary identity of the estrogen receptor was displaced by the discovery of the estrogen receptor beta. And this caused a minor endocrinological skirmish, that's what I call it, in its day, forcing estrogen receptors to be reclassified as either estrogen receptor alpha or beta. And this is when I began these nomadic wanderings in search of estrogen receptors in this in vitro model, moving in and out of petri dishes, in the shadows of transgenic politics, surrounded by scandals of scientific authority, exposed to gender, race, and class dynamics of scientists in the lab, and faced with the anxiety of protein otherness on many different scales. And that's what I call, basically, if you want to know what a day in the life of a feminist scientist is like, that's what it is. All this was worth it, however, because I knew that by participating in the production of scientific knowledge on the body, to working with the materials of the body, I would have a chance to bring together new feminist politics and this marginalized or minor literature in neuroendocrinology to form these new lines of inquiry. So many scientists, as well as feminist health advocates, for example, had suspected for some time that estrogens present in hormone-based therapies and contraceptives may be doing more than simply managing the symptoms of menopause or regulating ovulation. I was excited to participate in the production of scientific knowledge that examined the possible direct neurological impacts of estrogen and could perhaps help to address some concerns around the design and the use of estrogen-based hormone contraceptives and replacement therapies. So, the experiment itself involved proving that these neurons were actually creating the cDNA for estrogen receptors. And I recall the excitement that my colleagues and I shared when we first searched for estrogen receptors. We used molecular biology techniques such as subcloning and sequencing to find the genes for ER alpha and ER beta. And when we carried out these reverse transcriptase PCR experiments on these cells, we were able to easily find estrogen receptor alpha and beta in these cells. We also found not only the actual DNA being transcribed, but that there was proteins that were also being expressed in these GnRH neurons for estrogen receptors. And we used Western blot analysis to search for the expression of estrogen receptor alpha and beta, and both were found easily. And our results proved not only that GnRH neurons express these receptors, but that these receptors directly repress GnRH synthesis, and that was a different set of experiments. But the life of these experiments set me out onto this new line of flight and onto a series of interdisciplinary inquiries that continue to motivate me to this day. So since I conducted these experiments, it's really interesting. There have been more reasons to see that biologies are not fixed, and to appreciate that the value of moving toward new understandings of difference in our search for biological knowledge, it's crucial. It's been further reported, for instance, since I did this work, that estrogen receptor beta actually comes in not one, but 4 different orientations or isoforms, including ER beta 1, 2, 4, and 5. I don't know why there's no ER beta 3, but they decided to skip that. There's also a 3rd type of estrogen receptor that's been isolated, which is known as a G protein coupled estrogen receptor. It's also referred to as GPR30. And what's fascinating about this particular estrogen receptor is that a G protein coupled transmembrane receptor, it's not like the usual nuclear steroid receptor, which is what estrogen receptor alpha and beta are. When something actually is a G protein coupled receptor, the action of it or the effects of it can be felt a lot more quickly. Whereas when something is a nuclear receptor, the change that occurs is happening at the genetic level. So it takes a lot more time to actually move through the process of transcribing a piece of DNA, producing the mRNA into a protein form. So in GnRH neurons, which have been found to express this G protein coupled receptor, it means that estrogen not only regulates that nuclear transcription mechanism, which happens on a time scale of hours, but estrogen is also capable of eliciting rapid excitatory membrane initiated actions, which can take effect in seconds or minutes. So this membrane bound estrogen receptor can directly modify GnRH neuronal activity, and it can trigger several different signal transduction pathways that are not affected by the traditional ER receptors. So once again, this discovery of a membrane estrogen receptor may be of particular interest to feminist health and reproductive justice advocates, as these rapid and direct effects can explain kind of those long observed side effects that women have, such as headaches, a racing heart, dizziness, loss of appetite. So there are all kinds of other effects that can be happening through estrogen. So the discovery of these receptors and the further discovery of these G protein coupled receptors serve as opportunities, I think, for feminists and molecular biologists, not to just, talk about contraception and the importance of the pill, which is indeed important for reproductive justice advocates, but also kind of start exploring other objects of shared perplexity. So although I did not have the vocabulary at the time to express myself in this way, I knew that I had witnessed a kind of ontological rupture that had moved my understanding of matter from one of fixity and stasis and being to one of flexibility, change, and becoming through this experiment, through the life of this experiment. It was as if one night I had gone to bed with a materiality in which estrogen receptors could not be brought into closer proximity with GnRH neurons. And the next morning, I woke up to an alternate materiality with new sites of play and unexplored biopossibilities for these biological matters. So the life of this particular lab experiment continues to shape my efforts in theory making, beginning with my efforts as a scientist trying to become a feminist scientist, to a feminist scientist trying to become a women's studies scholar. This lab experiment has encouraged me to slip and grow in the midst of many disparate fields, and has extended itself into an immensely productive experiment in interdisciplinary scholarship. It has provided me with the impulse to bring together scholarship that is typically separated by disciplinary distinctions, such as the humanities and the sciences. Truly interdisciplinary work takes time, and I've learned that it's full of failures, and it can often leave everyone concerned unsatisfied. And as excited as I am to bring this interdisciplinary conversation forward, I'm fully aware that in my attempt to, write this book for multiple audiences, I may have in fact written a book that is legible to no one except myself. And even that is not guaranteed. What can I say? It's been an experiment. All right, so I'll end there. And, I'm happy to share a little bit more about that last slide that I showed. It's an NSF grant that I recently had, and where I was able to bring reproductive justice advocates together from Sister Song and Spark, which are located here in Atlanta, with molecular neuroendocrinologists who are looking at sexual differentiation in a way. They call it sex differentiation, but what I think they're looking at is what feminist theorists would call sexual difference. And I'll end there.
Thank you so much. That was amazing, and quite a journey that you've taken us on, Professor Roy. With 2 years of basic medical science training, right, many years ago, helped me to understand. So as a poor sociologist, then I sort of think that I managed to get by. So I mean, I want to just, I'm going to flag up a Zoom clap there, and I'm sure that other people might like to also clap, but my job now, it really is to convene and to curate the questions. So if people do have questions, could you perhaps just use the chat facility? You can make yourself visible on the screen by clicking your video, your open video. You can keep yourself mute if you wouldn't mind. But what I'm inviting basically is for people to just use the chat to say, I have a question or question, and then I'll come to you in due course. I've got a question from Emily, but I'm actually going to just take the privilege of being here to ask you a question myself, which is to ask, I mean, I found going back to Bose and the work on plants, I've just become very interested suddenly in trying to think about what else do plants do apart from those things which we make them do. But I was wondering whether, from your point of view, I think your talk indicates that we can learn an awful lot from the non-human. But do you think there's also a danger that we impose an anthropocentric perspective on how we start to understand plants or cells or receptors and create things in our own image, and that might possibly get in the way of true multidisciplinarity? Or is that an impossible resolution?
I think you're exactly right. There is always that danger, right? Because we are seeing it through our eyes, our experience, our language. But I think that the other side of it is also true, that we are being pushed and changed by our encounters with the non-human. And when language fails us, which I think happens a lot for those of us who are working in labs or not in labs, when we are ourselves pulled in a direction that we don't know where this is going, we've not been able to, we haven't experienced this before, we lack a language for it. I think that's the experience, that's the type of animal, human, matter, those kind of connections that we want to pursue. I'll talk about Bose, but I first want to talk about, like, one of those pauses that was created for me, and I talk about in the book, is I was actually being kind of, my own circadian rhythms were being controlled by the GT17 neurons that I worked with for 4 years. So part of my job was to measure the synthesis and secretion of GnRH, and that happened in a pulsatile manner in these in vitro cells. And because my part of my project was also looking at melatonin, which controls circadian rhythmicity, my own circadian rhythmicity was actually following for 4 years these cells. I had to withdraw samples from these petri dishes every 6 hours. So I would do a midnight sample, and then I'd be there again at 6 a.m. to do this. And so and one day it was one of those kind of like, who's doing an experiment on whom at this point? Are the cells working me, or am I working the cells? So that was one of those pauses. But to go back to Bose's work, I think we do run the risk of anthropomorphizing, but I think we also don't have a choice. And so if it gets us to a place where we have lost our language, lost our skills, and that's where we want to head. And I use this work actually, Getting Lost, in some kind of the other feminist STS work that I use, that's the role of the feminist scientist, is to make sure you're going down a road where the question you've posed no longer makes any sense, because I think that's a good thing. I think it's a good sign that you're lost in this world, you're lost in that lack of language.
Thank you very much. I'd again like to encourage people to turn on their video now so that we can actually see each other. But let's go to Emily for her question, and then I'll take people as they are inviting.
Nick, should we go to Greg, because he's got some questions and everyone's probably heard enough of me today?
Sure. Well, we'll come back to you, Emily.
Okay.
We'll fit you in, I'm sure. So, Greg, yes, please. Would you like to turn on your sound and ask your questions, or one question at a time, maybe?
Okay, yeah, perfect. I'll ask two, and if either of them are particularly boring, you can just ignore them. And yeah, so thank you for the talk and also for the book, which I really enjoyed. So the first question is, I'm currently reading Sarah Ann Wiley's book on fractivism, which I think is a kind of allied project in some ways to your own. And I'm kind of taken with her STS in practice and the way that she, in a sense, like develops new institutions of science in order to realize some of the emancipatory goals that I think probably you share some of those agendas. So the question now, I guess, is like, to what extent is this particular mode of science that you're striving towards, is that possible within existing institutional frameworks? And then the second question, which is totally different, is when I was reading your abstract, one of the first questions which kind of popped to my mind was kind of, oh my God, this kind of, if I just pull your phrase up, sorry, the physiology of response, particularly in the relation to reproduction, was around the kind of molar project of like abortion politics, and how kind of like imparting a responsibility upon particular individual cells in that context could be maybe in the context of the science wars, taken in a particular direction and used to further particular emancipatory goals. So I just kind of wondered there about how that kind of intersection of those molar and molecular feminisms might play out in a kind of situation like that. Yeah.
Those are great questions. But before I start, I have a question for you. How did the pasta turn out?
The pasta's awesome, thank you. Yeah.
Okay, all right.
The pasta is in the fridge.
So for the first part, how do you make these changes when you're in an institution? And I think the last 20 years of teaching undergraduate students has shown me that it's not easy, but there's something happening. Those undergraduate students who are planning on becoming scientists, we're preparing them to become scientists, are coming with kind of a understanding that science and society are not separate. That alone, right, seems like an accomplishment. I don't know who did it, but kudos. But these kids, and I will call them kids because I have college-age children myself, that they are aware of social justice issues, particularly what's been going on in the last 4 years here in the States. They are coming to this work knowing that what they do in the lab matters, how they communicate matters. And so picking those projects actually that align with some of the kind of ethical questions that they want to keep asking, and that cannot be contained by a 3-hour, once a year mandatory ethics training, on a computer. They are learning to live and inhabit in a space where those questions get to live side by side with their inquiry. So I'm seeing that happen. And, I will say that in the last 5 years or so, the number of double majors of students who are doing sociology and NBB, or women's studies and NBB, which is the neuroscience and behavioral biology, it's gone through the roof. And so, maybe it's too idealistic, but I think that there is something happening here. And I think this is what, a lot of feminists like Haraway were hoping and thinking, that this moment is coming, right? It is kind of maybe a product of the science wars, where, people were put into these two kind of camps, but there was definitely some kind of work and overlap and communication that had already been in progress, particularly in feminist science studies at that time. So I'm hopeful. And I do think you have to work within the institution. If you can do it separately, that's also amazing. But my whole idea has been, it's kind of a viral strategy that, you got to, and this is horrible to say it in time of a pandemic and COVID, but that it has been a feminist strategy as well, right? To kind of think like the virus and to enter and to use the machinery of the system to then get to a different end goal. So that's where I stand on that. The second thing about physiology of response, you're exactly right. No one said that this was going to be the way that we all want it to be. there's all modes of response that can happen, all kinds of lines of flight. And Deleuze and Guattari know that as well, right? So for reproductive justice advocates, if I think about that work, to use that argument of a physiology of response in the way that we can say, okay, well, now estrogen receptors, we want to develop a new contraceptive that maybe takes this into account, and so it won't be as harmful to women's bodies who use that. It's also opens up the conversation of a physiology of response between the fetus and the world, right? That, you know, that's a relationship that you, like, how can you ignore that? So what we have to, I think, also deal with in this is the kind of the ethics of killability, right? That and reproductive justice advocates, it's not about reproductive rights as such, but it's about a justice, right? That some lives, they know because of the eugenic past in the States here particularly, that some lives have been allowed to live and some not, right? So I think your question opens up a really rich, like, multiple areas, but I guess that microphysiology of response, it's not just for the things we want and we think are okay and good, it's for all of it, right? And we have to deal with that.
Thank you. Emily, would you like to ask your question now?
Yes, thank you. Thank you, Professor Roy, for such a wonderful talk. I'm kind of in awe of your experience in the laboratory sciences and kind of how brilliantly you weave this kind of informed perspective with feminist projects and in developing these, what you call, new lines of flight. And I'm only trained in social science, and I'm doing some research about experiences of the hormone HCG. And I'm wondering, like, how might those without scientific training like me gain some of this kind of similarly informed perspective? And even to what extent is this necessary? Like, what other sources of kind of biological knowledge can kind of be drawn on beyond laboratory experience?
Well, that's exactly it. You know, so after you know, I started training in women's studies, and I was exposed to more social scientists and those with expertise in feminist theory. This was my own project. Like, how do I build a kind of a dialogue and communication? So what I have found, that no matter what your project, so if you're working on HCG, first of all, I want to hear all about that also. That sounds great. But it's to partner, right? And that's been the most kind of, I think, unanticipated joy of this career that I've, you know, I didn't plan to do this work, but it has been the joy to make those connections with scientists, with activists, with you know, anybody who has kind of is a stakeholder of this knowledge. And sometimes there are people that, you know, you wouldn't necessarily want to collaborate with, but it's important to do so. So you know, where I always begin is I do a PubMed search, and I look at, you know, what are some of the earliest papers where the term even HCG came up, right? How did they think about it? And I always look for those minor stories that kind of get let go. So in this system of peer review, right, what we have is that each time a new wave of research happens or a new finding, a lot of things kind of get let go because it becomes kind of the dominant part of, like, oh, this is what's so cool about this molecule now. And this is how it can be applied. And this actually goes back to what Nick had mentioned about the utility of, you know, plants. We usually only think about it in terms of its utility. What else is it doing? And, you know, those stories are, something will always be embedded in a paper somewhere where someone caught something about HCG that they didn't, you know, nobody followed up on. And those are the stories that I love to go after. You know, I mean, we could talk more about this, but PubMed, I mean, the research is there. And I think scientists themselves are asking those questions that are, if you listen to it or you have that toolkit, you can hear that they have an ethical quandary themselves. They phrased it in a certain way so that it doesn't seem like, you know, that's the, you know, the important part of the paper, because they're not supposed to be asking those questions. but I'm always drawn to the statements that seem a little bit kind of, there's an open door here, that they're inviting us to discuss this.
Thank you so much. That's brilliant. I love that idea about minor stories. So thank you. Oh, Nick's on mute. I think he's.
Yeah, we've got a couple of minutes left, so I'm not sure if anyone just wants to jump in quickly, but while we're waiting, and I'm going to use every minute of your time that we can. Can I ask you one question? You had one slide, and there were a lot of statements which said something with the word "we" in it, and I think one of them was how are we to encounter matter? And I guess that maybe there's a question about who the "we" is as well here that we have to interrogate more critically.
That's great. The "we" here, I think is the feminist scientists working by themselves in a lab, who maybe knows that there are collaborators and co-conspirators out there that will get their project going forward. So I do imagine that there are people in post-colonial studies who are aware of these critiques, but who would maybe, for example, argue that we never had the luxury to have this flight from nature, right? That certain bodies have been tied to it, and how do we communicate what we've been doing? And that's the "we" that I think. So I think it's the people within, it's a multiple "we." So it's those who have been marginalized within science itself. So this is where you can think about DEI issues and the whole horrible pipeline metaphor that I hate, but you know what I mean? Like the people that get lost in science or somehow leak out. The "we" is those who are, even within feminist theory, there seems to be kind of a very kind of like a norm that's been established. And in the book, I do talk about kind of this very American-centric kind of understanding of biology and matter, which a lot of European feminists have been saying that there's been a misinterpretation of Simone de Beauvoir's work to think about materiality and process and biology. So I think it's that "we" as well who have been left out of that dominant feminist theory conversation. And a lot of new materialists who, I think they're trying to do something different. And it's not all about kind of the charge that feminism has left matter behind. That new materialism is also about unearthing the work that has been going on for a long time, whether that's through historical materialisms, through standpoint theory, and to look at which lives and which labor has counted in our theorizing. So those are the different "we's."
Thank you very much indeed. I think back to you, Emily, to wind us up now.
Yes, great. Well, thanks, Nick. So I just wanted to close by giving our thanks to Professor Deboleena Roy on behalf of the organisers and the audience as well for a wonderful and eloquent talk that's given us lots to think about. So thank you. And thanks those who asked questions as well. And I also wanted to acknowledge the support of the BSA events team in hosting this event. So we don't currently have any further seminars planned in this series, but due to their popularity, we'll definitely discuss this, and also the possibility of rescheduling Angie Willie's talk, which unfortunately had to be cancelled a couple of weeks ago. So look out for updates on the mailing lists. So thank you everyone again for your participation, and to Professor Roy for such a fascinating talk. Thanks very much.
Thanks everyone.
Thanks.